and a little bit about the prevalence of this disease. So in the United States, there are about 500 to 1,000 new cases of PAH each year. And this disease is most common in women between the ages of 30 and 60 years old. So this leads to an interesting paradox around estrogen that is being investigated today. So it is known that women are 4 times more likely than males to get this disease. However, they are also way more likely to survive the disease than the men that get this disease. So this brings questions around the role of estrogen, which is the female sex hormone, and its contribution to heart failure and disease. And then the reasoning behind this estrogen puzzle is very unclear. And so some past research has shown that estrogen has a protective effect on the heart, and others have shown a disease promoting effect. So currently future research is still needed around estrogen's role in pulmonary arterial hypertension. And so I'm interested in determining the role of estrogen in PAH, even deeper the role of estrogen on the right ventricle. So it has been found that right ventricular function and morphology is an important predictor for the outcome of PAH. And so throughout the progression of the disease, the right ventricle remodels. So an RV remodeling can refer to changes in geometry, pressure volume relationships, wall thickness, and the function of the heart. And so past research has shown that the RV initially adapts. So these are showing the adaptation of the right ventricle. And it does this by increasing muscle contractility and wall thickness. But eventually this will lead to severe decompensated right ventricle failure. And that is that is what happens when the heart fails. And so with the decompensated RV failure, this leads to reduced cardiac output and wall stiffening. And so there's a point in time in which the RV goes from this adaptive remodeling into maladaptive remodeling. And we're not quite sure what that exact point is and why it decides to change from this adaptive state to the maladaptive state. But once it reaches the maladaptive state, there's no return to adaptive state. And so the maladaptive state leads to right heart failure and then premature death for patients with PAH. And so I'm hypothesizing that estrogen plays a major role in the remodeling of the right ventricle, leading to an overall protective effect against right heart failure. And so I aim to quantify the hemodynamics of the right ventricle through the progression of PAH using a rat model and to identify sex-dependent effects of estrogen in the remodeling and mechanics of the right ventricle. So this is just an overview of what I've been doing this past quarter in my first year at UCSD. And so I'll go into each box specifically. So box A shows the animal interaction. So we are using a rat model, and we use a Sugen hypoxia protocol to induce PAH into male and female rats. And for female rats, there's going to be 2 groups of female rats that we have been using. So we have overectomized female rats and then non-overectomized female rats. And so this is hoping to represent menopause, so like premenopause and postmenopause females. And so the rats that we want to have PAH, we will inject with a vascular endothelial growth factor receptor and then inhibitor. And then this is followed directly with 3 weeks in hypoxia. So this is just injecting the inhibitor and then the 3 weeks in hypoxia, which is 10% oxygen levels. And so this protocol has been used, and it's widely used to induce pulmonary arterial hypertension, and it has been found to mimic common clinical features that are found in humans, such as plexiform lesions. And after the 3 weeks in hypoxia, we take the rats out, and then we leave them in normal air conditions where the disease will continue to develop for a designated number of weeks. So we test at different weeks throughout the progression of the disease. And once we're doing surgery, which I'll talk about next, but in order to determine that PAH was successfully induced, we look at the mean pulmonary arterial pressure, which should be above 20 millimeters mercury if in a PAH patient. So box B shows the terminal surgeries that are performed at the different stages of the progression of the disease. So my role in this has been assisting in the setting up of the surgical bench and all the tools, and also the cleaning up of these surgeries. And then during the surgeries, I am taking notes of what's happening. And I also continuously monitor the heart rate. And so if the heart rate goes too low or too high, I adjust the isoflurane level, which is what we use for anesthesia for the rats. And then I do any additional assistance that is needed while Dr. Valdez-Hasso is performing the surgeries. And so you can see these things are representing catheters. So a specific pressure volume catheter is inserted into the right ventricle of the heart. And this allows for in vivo blood pressure volume relationships to be measured. And the goal of this is to investigate changes due to pressure overload from PAH. And so box C is showing an example of a pressure volume loop. So this we'll get similar loops to this using the pressure volume catheters during the surgery. And so pressure volume loops are the gold standard for measuring direct real-time cardiac function. And so the PV catheters record the change in volume and the change in pressure during one heartbeat or one cardiac cycle. And so then this is just plotted pressure versus volume to form these PV loops. And so once the rat's heart rate is stable and the catheters are all showing good readings from our program that we use, which is like lab chart reader, then we will record the PV loops. So after each steady state measurement, which is when the heart rate is stable, we will also occlude the inferior vena cava, which removes the preload from the PV loops. So these occlusions are just the gold standard method for obtaining load independent measures of contractility. But for what I'm working on right now, I'm using the steady state PV loops and comparing those. And so what these PV loops do is they help us to identify the differences in hemodynamic metrics such as heart rate, end diastolic pressure and diastolic volume, and systolic pressure and volume. And then we can use these values to calculate stroke volume, ejection fraction, and cardiac output between the different animals within a certain group and the different animals between groups at the different stages along the progression of the disease. This last box D, shows the steps that follow the terminal surgeries. So after each surgery, the right ventricle of the heart is harvested, and it's run through a planar biaxial testing protocol, which you can see an example of like right here. And so throughout this past quarter, I've helped with running these biaxial tests. However, I have not helped with the analysis of these results, so I am not familiar with those yet, but I hope to start analyzing the results soon. And then also histological analysis are done to look at the composition of the tissue and the collagen content, etc. And I have not done any histological experiments yet, but I'm hoping to also do that this quarter to tie all my research together. And so what I'm currently doing is I'm currently been focusing on the pressure volume loops for each subset of animals with the goal of finding patterns to describe the differences in the heart's function for males, non-overectomized females, and overectomized females. So on this screen right here are all the current loops for control for male animals. So the top is male control animals week 6, and then the bottom is male SUX animals week 6. And so you can just see the difference. So the control animals, the loops are much more similar to each other with the rectangular shape, and there's less variability, which makes sense because hearts relatively function the same for healthy individuals. However, the disease affects all of these rats very differently with their heart's function, so we get a wider range of PV loops. So this is for male animals, and this one is for non-overectomized animals. So these are just normal females with their ovaries still intact. And again, you can see the control loops up top, and then the SUHEX week 6 animals on the bottom. And this slide shows the overectomized PV loops for control animals on the top, and then SUHEX week 6 animals on the bottom. And we do have less animals of for these overectomized, this overectomized group, but as we add more, I'll be adding more to my graphs. And then just some observations that I've found so far. So these are all just from looking at the graph. So these numbers are averages, and my descriptions are just from visual interpretation. But something that I've noticed is that the ESP, which is end systolic pressure, end systolic volume, end diastolic volume, and end diastolic pressure for the control animals are all less than for the SUX animals. So for example, the end systolic pressure for the controls are all relatively around 25 millimeters mercury. But then when we look at SUHEX animals, it's a relatively about 65 millimeters mercury. And so the same is the case for end systolic volume and diastolic volume and end diastolic pressure as well. It's relatively higher in the SUHEX males. And then also for the shapes, the controls are more rectangular, and then the SUHEX are more pointy on top. So if we go back, we can see the rectangular shape for the controls. And then for all the SUHEX animals, there's more of a point on the upper left-hand side, which is something I've noticed for every group. These ones aren't all as close, but they're mostly rectangular.
So one thing you see there, Kristen, that looks like there's a trend towards the SUHEX females having lower pressures and lower end systolic volumes. In other words, being closer to controls than the males and the overectomized.
Yes.
So that would go along with the idea that there could be a protective effect of estrogen.
Yeah, exactly. And then I've also am looking at isovolumic contraction and relaxation phases. And so I found that the relaxation phases are relatively all vertical, but the contraction phases are more slanted. So just going back, so the relaxation's more vertical, but then there's more of a slant in some of these SUHEX animals. So I'm currently still working on dividing up these PV loops into different categories in order to attempt to determine like similarities and differences between the groups and conclude what's the main differences. And for example, like one way that I'm separating them is, so these are all the control non-overectomized animals. So it's every week that we have. And then I separate them to early weeks and then late weeks down here. And so I'm also going to narrow them down between each week by itself. So have week 1 PV loops and week 2 PV loops, etc. And so I'm doing this in order to try to see if we can expect a specific range of values for the PV loops for a given group. So like if we're given a week 6 SUHEX female, can I expect, or can I like determine what the values for the pressure and volume would be before actually checking them with a PV catheter? And so my future plans are to dive more into the estrogen component of the RV remodeling in PAH. So I want to determine the baseline amounts of estrogen in each animal and then the estrogen levels throughout the progression of the disease. And something that I've been recently looking into is the estrogen levels during the estrous cycle of rats. So the estrous cycle of rats is just like the menstrual cycle of humans. And so there's different levels of estrogen in the body at these different times. So I'm curious to see if we do surgeries at specific stages of the estrous cycle, if that affects how much estrogen is in the rat and therefore affects the heart's function. So that's something I'm going to try to look into more this quarter. And I also want to relate just the estrogen levels to each PV loop and the estrogen levels to stress strain measurements using the planar biaxial testing. And something else that I've thought about is determining if estrogen is the main factor that we should be looking into, or if there's something else that's influencing the mechanics of the heart in females versus males that is also worth looking into to get a better overall idea. And so that's pretty much, these are just some references, but that's it for this presentation. And I hope to like narrow down my project topic in this quarter. And then so next time I present, I'll have results and a better idea of what I'm going to be doing.
Thank you, Kristen, and thanks for being sort of willing to give a talk, when you still don't have only just new results. I really encourage students to do that. Are there any questions for Kristen?
Yeah, that was really cool. One question that I had, and this might be kind of way outside the scope, but in terms of trying to categorize some of the PV loops, have you thought about trying to use any kind of machine learning algorithms or neural nets or something? I know that a lot of times for other kind of electrical signals, we can classify them using neural networks or something. So obviously a PV loop and a neural signal are not the same, but I'm not sure if you've thought about kind of other ways to categorize that beyond qualitative kind of observations.
No, not yet. That is a good idea though. Right now I was more just trying to average the loops, maybe to get all like 1 week into like a average with like a area of like a confidence interval, but that is a good idea to look more like quantitatively.
So that does bring up an interesting thought, because the nice thing about the PV loop is it's state space, right? You take time out, so you get to see the sort of steady state hemodynamic behavior, but not everything is steady state, and there are temporal components too. In fact, you point out that spikiness at the at the end of the hypertensive loop. I mean, that's really a function of the shape of the pressure waveform, which in turn is affected by the mechanical properties, resistance and capacitance of the arterial system. So you do lose some information by taking time out. And I mean, the pressure volume diagram is very, very valuable, but it might be interesting to, if you were going to do some more sort of machine learning type of analysis, to look at the time course of pressure and volume, where you still would have the pressure volume relation implicitly, but you'd also have the pressure time and volume time. And then, the interesting thing is, you have controls where they all seem to start out pretty similar. And then you have an early phase where they're all relatively compensated. And then sometime later, there's some loss of function. I'm not sure that you're getting out far enough that they're really failing or decompensated, but there's definitely a decline. However, it's probably different between the groups. And what might be really interesting is to see if there is something in the early signals that predicts the later ones, right? In particular, it would be interesting to see if, because what you saw, how you said you saw an increase in end systolic pressure, you also saw an increase in end systolic volume. So as the pressure goes up, the volume will tend to go up. The more it goes up, the more the volume will go up. But then you also saw an increase in diastolic pressure and volume, and those are more indirect. On the other hand, there's nothing to say that those mightn't have come first. So it would be really interesting to see if you could find some predictors from the early time points of the later ones, or of the early and intermediate to the later, that might give you a clue as to what's driving this. Is it really the increase in systolic pressure causing the end systolic volume to go up? Or is it maybe the fact that the end diastolic volume goes up and that increases the end diastolic pressure, and that in fact could be the driver of the adversary of the decompensation? I don't think anyone knows. And I think, you don't have enough data to do deep convolutional neural networks, but you probably do have enough to use some sort of algorithms. So that might be interesting.
Yeah. One thing that I've noticed though is, so each rat, we only look at the PV loops on the day that they, of their surgery. So we can't really compare like that rat's early week PV loop to that specific rat's late week PV loop, which is something that, because each rat is different. So I'm not sure.
Yeah, that's a big limitation for sure.
Yeah.
Are there any other questions for Kristen? If not, I wanted to comment on one thing, because it just crossed my mind when you said you had a background in applied mathematics, and we're doing now sort of learning the physiology and biophysics and biomechanics. Well, you may not know this, but this program is called the Interfaces Graduate Program, and it was called Interfaces by the Howard Hughes Medical Institutes that created the initial grants that initiated the 10 interfaces programs around the country. But they didn't come up with the name interfaces. They actually borrowed the name from the Burroughs Wellcome Foundation, from the Wellcome Trust, that had earlier created a program called Interfaces that was mostly a postdoctoral training program. And in fact, we had one here at UCSD many, many years ago called La Jolla Interfaces in Science. The interesting thing about that Interfaces name is what it was actually about was exactly what you're doing. It was actually all about bringing students with physics and applied math and engineering backgrounds and no biology into biomedical sciences. Nowadays, you know, things are more multidisciplinary, but I couldn't help being reminded of the origins of that Interfaces name. So, you know, you're an Interfaces classic. Okay, well, if there are no more questions for Kristen, thank you very much again.
Thank you.
Now we'll switch over to Marcus, who I think is probably going to be talking about 2-deoxyadenosine triphosphate.
Exactly. Yep. And hopefully everything looks good to you all. And let me just move my video of everyone else. And yeah, so hi everyone, I'm Marcus. Here's a quick background about me, since a lot of you I haven't met, at least not in person, but I'm a third-year PhD student in the bioengineering department, and I got my bachelor's in chemical engineering from CU Boulder, and I moved to San Diego in the fall of 2019. And my research interests are in molecular muscle mechanics and computational chemistry, and kind of building multi-scale models from that information kind of interface. So a quick outline of kind of everything that we're going to go through is a little bit of review of cardiac physiology. Since I know we have such an interdisciplinary group, I want to kind of remind people some of the molecular underpinnings of the muscle contraction. And I want to talk about hypertrophic and dilated cardiomyopathies and some of the causes, and some of the approaches that I've been using, using computational methods to try to predict both therapeutic effect and disease effect and different mutations, and how we might be able to treat these diseases with datp, as Andrew mentioned. So a quick refresh into going from the organ all the way down to single molecule physiology. So right, we have our heart that everyone is familiar with, and the ventricular walls are obviously essential for getting the blood pumping out of the heart and to the rest of the body. And our cardiomyocytes are these striated cells that are a little different from skeletal muscles, but still very familiar in the sense that you have your sliding filament theory, where you have myosin heads that slide past actin filaments. And it's this progressive Crossbridge cycle that leads to actual muscle motion. And so at the heart of it, we have the single myosin molecule, which is a molecule that I've been studying in depth. And you can see using cryo-EM, I think this is what this image is from. You see this kind of Z-band, familiarly the M-band. And, you know, if we can go from this cartoon to this image, it's a nice demonstration. So ultimately, at the core of the muscle contraction, we have an influx of calcium, along with a lot of other ions and a lot of other electrochemistry happening. But this triggers the movement of troponin and tropomyosin to allow myosin to activate and bind to the actin filament. And so we can go from our relaxed to our contracted muscle filaments, where the filaments slide past each other, hence the name sliding filament. And at the core of this, we have what's called the Crossbridge cycle. And so this is a cyclic motion of myosin, what repeatedly goes through ATP hydrolysis, where initially we have myosin and actin are separated, as you can kind of see in these cartoons. We have this attachment phase, which is what I'm going to focus on most through this talk. And then we have what's called the power stroke, and that's a mechanical motion of the myosin head, where you can see this kind of neck region goes to a different location, and that's what generates power. We have a release of phosphate, release of ADP, and then a detachment phase, and then a recovery stroke, where we kind of reprime our myosin head to contract and generate force. And so if we want a nice little animation, we can very quickly watch this happen, hopefully, where we have our filaments, we have our myosin heads. You can see the calcium binds the troponin, that moves the tropomyosin, which then allows the myosin heads to activate. And hopefully this will zoom in a moment. Then we can, so yeah, we have our previously hydrolyzed ATP, which is now ADP and phosphate. It can now bind because the tropomyosin has moved, and then we can get a ratcheting motion, which should happen as soon as the phosphate is released. And this is something that's actually still up for debate, is exactly how the power stroke happens in regards to phosphate release, but that's not something that's super important. But anyways, you can see we have this mechanical motion. The myosin heads will dissociate as a new ATP comes and binds. And then after hydrolysis, we reprime the myosin head and we can repeat the cycle. So there you go. We're kind of back to where we started. And now you are all experts with the Crossbridge cycle. So moving back to kind of the whole organ, the whole heart, and diseases associated with it, two that are extremely common are both dilated and hypertrophic cardiomyopathy. And so as the names kind of suggest, you can imagine what might happen to the ventricles. And so during dilated cardiomyopathy, the ventricles become significantly larger, the walls thin, which leads to overall muscle weakening. And in severe cases, this leads to heart failure. There's a variety of causes that can cause dilated cardiomyopathy, ranging from lifestyle, obesity, arrhythmias, but also genetics and different mutations to proteins within the cardiomyocyte. And hypertrophic cardiomyopathy, as you might expect, kind of is the opposite, where you have the walls thicken significantly, the ventricles become smaller, and the ventricles also stiffen. And this can lead to, again, heart failure or sudden cardiac death. A lot of times we hear about this with athletes. And again, this can be caused more generally often by genetics and mutations. And so people have done a lot of analysis to try to understand what kind of mutations are causing hypertrophic and dilated cardiomyopathy. So again, here's kind of our representation of the sarcomere. And so titin has been associated with dilated cardiomyopathy. The myosin head and tail have, primarily the head though, has been heavily associated with both hypertrophic and dilated cardiomyopathy, myosin binding protein C, which isn't pictured, and filament C. These are all different proteins that are important for cell contraction that have been kind of associated with these cardiomyopathies. So I'm going to focus more specifically on the myosin head and try to understand how mutations to this myosin head cause diseases, as well as how we might use therapeutics, DATP, to treat dilated cardiomyopathy. And so kind of there's our quick background of cardiac physiology. So DATP is a near analog of ATP. And literally the only difference is you replace this hydroxyl group with a hydrogen, and we now have DATP instead of ATP. And this is something that's naturally occurring in the body. It's actually what's in DNA. It's the deoxyribose as opposed to just your ribose as an RNA. And so this isn't anything synthetic or new, but when you increase the concentration of DATP in cardiomyocytes, it's found to increase the contractile force. And so I have a couple of figures here from the Rainier lab, who's been studying DATP for probably longer than I've been alive at this point. And so you can see that the steady state force with DATP is significantly higher. The relaxation is faster with DATP. And if we look at the normalized length, we see that this R1R2, that's enzyme that converts DATP to, or ATP into DATP. So this is kind of our DATP case. You can see that it contracts further with DATP as compared to our no treatment. And the GFP we can kind of ignore, but that's just a control since we're using viruses to inject R1R2. So I want to study DATP kind of from the source and moving outwards. And so that means kind of looking at myosin and how DATP can be used instead of ATP. And working backwards, we need myosin. And so this is obtained through a lot of advanced crystallography that I'm not an expert in, but people are fortunately very good at this, where you're able to crystallize your protein, use X-ray diffraction to then basically rebuild the molecule, and you can figure out exactly where each atom is in 3D space. And using these models, we then can start to carry out simulations, whether it's molecular dynamic simulations or Brownian dynamic simulations, which I'll talk a lot about, and ultimately understand structure and function relationships better, and maybe design new therapeutics or understand how therapeutics work. So this has a lot of applications. And so kind of where I'm focusing right now is in this computational chemistry realm and scaling that up. So returning to these mutations that cause DCM, there's a couple that I've been focusing on lately, and there's this S531P mutation. So we're going from serine to proline, as well as this A22T mutation. So you can kind of see this just barely in this yellow behind this beta sheet, this A22T mutation, which is relatively close to the nucleotide binding site. So that's why we're interested in studying how DATP might affect this mutation and overall function. And this S513P mutation is on the actin binding surface of myosin. So if we now have our kind of actin filament, you can imagine it goes along diagonally across the screen. There's just a few monomers of it. you can see that this mutation is right on that surface that interacts with the actin monomers. So because of the unique locations of where these mutations are, and they've previously been identified to cause DCM, that's kind of why I'm looking at them and trying to use them as case studies to better understand how DATP might act as a therapeutic and how other treatments might influence myosin, or how other mutations might influence myosin behavior. So kind of as an overview as to what I've been doing, I've been starting with a myosin crystal structure. And then I have had a collaborator at the University of Washington, Matt Childers, has been carrying out these MD simulations. And so we've been carrying out these 4 different conditions where we have just our wild type with ADP. And I'll be saying ADP a lot instead of ATP, and just know that that's because it's in the pre-power stroke state and the ATP has already been hydrolyzed. So similarly, we've been simulating the wild type myosin with DADP, as well as these AT2T and S513P mutations in the presence of ADP. And from these simulations, I've been using some clustering algorithms to try to reduce all of this data that you get from an MD simulation, because you have an enormous amount of data, into something that's a little bit more palpable and more informative. So I've been constructing these 15-state Markov models that kind of showed differences in myosin structure. And then I've been incorporating these into Brownian dynamic simulations. So when we go from MD to Brownian dynamics, or BD, we're kind of increasing the scale, and now we're able to look at things that happen on a much slower time scale than traditional MD, and we can actually look at protein-protein interactions. So I'll get into that a little bit more in a moment. And right now what I'm focused on is this association rate. So if we go back to the Crossbridge cycle, right, there's this Ka where first myosin and actin are not attached, and then myosin becomes loosely attached. And so this first step is what I've been looking at, and obviously there's several other steps in the Crossbridge cycle, but right now we're focusing on this association rate. So kind of talking in a little bit more about how this clustering and Markov model works is I'll start with our MD simulation. And again, this can have 10,000 or more frames where each atom is explicitly measured and you know where it is in XYZ Cartesian coordinates. And from this, I've been using a clustering algorithm to identify 15 different states that are hopefully representative of the myosin as a whole, because if I have 10,000 different frames, that's a ton of data to work with. And using something that's a little bit more representative of subtle shifts in the myosin is going to kind of be much more useful, especially if I'm trying to use Brownian dynamics in conjunction with this. And so this matrix right here, this heat map, basically shows how you can transition between different states. And so if you're in state 0, for instance, it could transition to a state 3, state 2, or a couple other states. And you can look at this across all of our 15 different states that we've generated. But ultimately, it's a little bit nicer maybe to look at this as a graph, and you can see here now very clearly how state 1 can transition to other states, state 7, etc. But most often, a state is just going to stay in its same state moving forward. And so that's kind of why you see this diagonal that's significantly brighter than the rest of the map. So comparing all of these different conditions, this is kind of just a comparison on a visual side, I guess. There's no specific metrics, but it is really interesting to note how different the transitions and the kinetics are of each of the different states, each of the different simulation conditions, whether it's ADP, DADP, the AT2T mutation or the S531P mutation. Most notably, I think it's surprising that both this DADP and S531P mutation have far fewer transitions into other states, and this diagonal is much brighter. And you'll notice that kind of correlates with some results later. Whereas the ADP and the DADP, there's much more kind of kinetic behavior where there's faster transitions between different conformational states for the both just wild type ADP case and the AT2T. So another way to kind of visualize this is just looking at whether or not there is a transition. So if we convert this basically from a gradient of a transition probability to a heat map, now if it's just any connection, we can visualize this. And again, the S531P stands out to be quite different from the others. And whether or not this is truly meaningful, I think is up for some debate, but I think it's interesting analysis nonetheless. So now moving to Brownian dynamics, now that we've kind of built these Markov models, basically Brownian dynamics is a simulation technique where you're using rigid bodies and electrostatic forces to try to predict how proteins interact or don't interact. And so ultimately, you're every time step moving your molecule a little step dx, and it's going to be a function of both the electrostatic and the Leonard-Jones forces, F, as well as some random vector W, which is based off of the Brownian motion. And so if we keep iterating this, we can watch as either molecules dissociate from each other or come together and form a reaction. And so kind of this is the backbone of Brown dye, which I know several of you guys have learned since we've taken the multi-scale interfaces class. One of the maybe shortcomings you could say of Brown dye is the fact that you need to set this reaction distance or this reaction criteria as far as how close the molecules need to come to each other. And so just kind of for an example, if we set our criteria to 20 angstroms and we say we need our molecules to come within 20 angstroms of each other to have a reaction form, you can visually see this is still pretty far. So previously we've been kind of looking in the 10 to 5 angstrom range in terms of how close the molecules need to be considered a reaction. Of course, since it's adjustable, it's a little bit more useful to just look at the range. And so all of these Brown dye results you'll see are going to be kind of on the spectrum, and we can compare the overall trends between different simulation conditions. And so one other thing to note is this is just one kind of Brown dye experiment. So you'll run several of these algorithms, several thousands, and from them then you can kind of estimate this binding curve as far as some trajectories dissociate and some form a reaction and do associate with each other. So now if we try to incorporate our Markov models with Brown dye, we can get a lot more information and start to compare and understand how datp is different from ATP as well as these mutations. And so each one of these curves shows the results from a set of Brown dye simulations, and each line corresponds to a unique state from the Markov models that I built previously. And so you can see, for example, this third state might correspond to this line. That's just kind of an example. And so each of these has 15 different lines, and you can see DADP has a significant general increase in terms of the second order binding rate is when you compare that to the ADP case. And if you look at the HE2T mutation, it's subtle, but it's a little bit lower than the wild type. And the S531P actually increases again from wild type. And so since I have all of this information from the Markov model that I've built in the transitions, we can kind of create a new super Markov model where we incorporate an additional 16th state into the Markov model. And so now we can transition between different conformational states of myosin. And then ultimately, there will be a final state where the myosin binds to actin, and that's kind of the end. So we can sample this randomly using Monte Carlo sampling, and then finally estimate kind of a generalized Brownian dynamics rate that is incorporating all of the kinetics of the MD simulation as opposed to just one specific frame. And so this shows in solid line this kind of Markov sampled rate for each of the different conditions. So again, the black is the wild type, and you can see that if we use DADP, we have a significant increase in the association rate. Then if we go to the AT23, which is the same as the AT2T, there's a subtle nuance depending on what type of myosin you're using, but again, the same mutation, we decrease quite significantly by about a factor of 2. Interestingly though, this is still a DCM mutation, but we have an increase in the fold change of the S532P mutation, the S531. And so this is just kind of this average fold change on the right, which is just basically a ratio of these solid lines between. And then the faint lines, you can see just the other states still. So there's an extremely wide range between different states. So I think a little bit more analysis needs to go into why this state is so much faster than, say, this other state that's significantly below it. And so digging into that a little bit, the actin surface that binds to myosin is predominantly negatively charged. And so understanding electrostatic changes on the surface of myosin is one kind of mechanism as to why we might see such different association rates. And so loop 2 is, sorry, a little bit out of order, but loop 2 is on the surface of myosin that interacts with actin quite strongly. And so we can look at basically how positively charged the surface area is of loop 2 and see if that correlates at all with the binding rates. And so we have the wild type ADP, the DATP, the AT2T, and the S531P mutation. And in general, these correlate quite well with the behavior that we notice with the association rates, where DADP has more surface area that's positively charged, has a faster binding rate, as we saw in the previous slide. Oppositely, the AT2T has a smaller surface area. And again, the S531P, higher surface area, higher binding rate. And so these figures, each one of these corresponds to a unique state in the Markov model that I've built. And then the size of the circle just corresponds to kind of how frequently we see that state. And again, so loop 2, which is kind of what surface area I've been looking at, is a disordered part of myosin. It's not an alpha helix and it's not a beta barrel. It just kind of flaps around all over the place. So it's been shown to have a pretty significant influence on myosin behavior. So that's why we chose that. And you can see that just barely in red. And that moves around quite a lot during these simulations. And actually, if you try to crystallize myosin, you won't even be able to see what the loop is because it's so disordered. And again, so here are those trends. And so kind of the one thing that I wanted to look at is whether or not there's a correlation between each different state in the binding rate. So as opposed to plotting the polar surface area on loop 2 versus each condition, we can now look at it versus binding rate. And so this general trend that we see on the graph on the left suggests, okay, higher surface area with polar surface area on loop 2 should probably be a higher binding rate. And ultimately, that's not the case. So for each of the different simulations, I've looked at the correlations, and so you can see that colored accordingly. So for example, the ADP wild type case, there's a subtle but non-significant negative correlation with loop 2 among its simulation. If you look at the ADP, there's an even stronger correlation negatively with increased loop 2 surface area and the binding rate. And then oppositely, both for the A22T and the S531P, there's subtle positive kind of relationships between the binding rate and the loop 2, but all of them are non-significant if you look at the slopes of any of these regressions. And I also looked at just an overall kind of all of this data and the correlation to understand if this truly is a good predictor, and this slope is also non-significant. It's almost significant, but I think that given the diverging trends, it's maybe not an explicit mechanism to really explain what's happening. So that's one of the next questions, is to really try to understand why different myosin behave so differently in the context of association to myosin, or sorry, to actin, as well as other steps in the Crossbridge cycle. So one thing that I've been working on recently is trying to build a more realistic Brownian dynamic simulation by kind of restricting the motion of this myosin head. So traditionally, Brownian dynamics, the two proteins are just kind of free floating in solution and can go wherever. But realistically, in a sarcomere, it's a very intricate lattice system, and the myosin head is tethered to the thick filament, and it's not going to go anywhere very far away. And the actin filament is obviously fixed. And so the goal is to basically restrict the motion in these Brownian dynamic simulations using a series of springs, such that we can look at longer timescales and better understand how myosin might bind to not only just one actin monomer, but possibly other sites, and try to understand maybe duty ratio as well, and look at detachment steps. So I think that moving forward, this could be really useful to understand this better. And people have previously created spatially explicit models of the sarcomere using similar spring setups, not using Brownian dynamics, but kind of we're drawing on inspiration from these models from Williams, understand a little bit more how myosin and actin might interact. So ultimately, there's a lot of additional questions to be answered. So I still would like to dig into a little bit more as to why the different mutations have such different diverging affinities for actin, and to understand if there's other features that might better kind of predict why it changes. So the myosin cleft is something that I didn't talk about at all, but basically there's almost this little jaw in the myosin head that can move open and closed as it moves through the Crossbridge cycle. Then ultimately, we want to understand how DADP can work as a therapeutic for these mutations. And so right now I've just been simulating A22T and S531P with ADP. And next we need to work to understand what happens with DADP. And so again, then I want to finish kind of understanding this spatially explicit model to restrict the myosin head motion. And ultimately, there's several steps in the Crossbridge cycle. So to look at dissociation steps or ADP release or phosphate release, these are all more difficult questions to ask, but there's additional software. So for example, Seeker, which has been developed by the Amara lab, can look at dissociation rates. And so this, I think, is something that I'm going to start to dig into a little bit more, and hopefully be able to build a more complete description of changes to the Crossbridge cycle using these in silico approaches. So with that, acknowledgements to Andrew McCulloch, my PI, Abby Teitkin for all of her help, and the rest of the CMRG lab, as well as Gary Huber, he's been the brown dye expert and really helpful, Matt Childers, who's done all this MD, as well as the interfaces training program. So can now take any questions, and thanks for listening.
Thank you, Marcus. We have time for questions for Marcus.
Great talk. This was really cool. So when you were showing your heat maps, it looked like the S531P case was transitioning less overall, right? And then the A222T is transitioning more, but then the S531P had a higher myosin actin association, right? So how do you kind of link those two things?
Yeah, that's a good question, and something that I hadn't really looked at recently. But so kind of what I'm thinking, at least if we look at these two heat maps, both the datp and S531P seem to be more stable in their existing states, as opposed to, like, you know, the diagonal is brighter, so they're not transitioning as frequently. And so I think that that might correlate to then being more favorable to bind to the actin surface. Whether or not that's true, I think I need to dig into a little bit more. But that's kind of my weak hypothesis right now, is the fact that it's already kind of happy where it is, and so it's fine to bind to actin. Although I guess you can make an argument that if it was less stable, then it would be more likely to bind. But that's something that I think I need a little bit more analysis using maybe energetics of binding to do that.
Yeah, that'd be super interesting.
I'm also sort of reminded of a little piece of interfaces history with your presentation, Marcus. You know, a lot of these methods for linking modeling at across different molecular and cellular scales were developed by various instructors and students in the interfaces lab course on multiscale numerical analysis for multiscale biology, which we started, 15 years ago. So a lot of the projects that the students in that class had ended up motivating new developments that ended up, being used for all kinds of research like you're describing. And you mentioned Seeker. So Seeker was developed by Lane Vitapka, and Lane was an interfaces student about 6 or 7 years ago in our class. And this quarter, he'll actually be teaching some of the molecular modeling, including Seeker, hopefully. So it's just, it's nice to see how the interfaces program is itself developing the new technologies that new students learn.
Yeah, definitely. That's really cool to come full circle.
Are there any other questions for Marcus?
I have one.
Yeah.
Marcus, could you explain a little bit more about the, I guess, the springs that you're adding to your Brownian dynamic simulations? I think I just don't,
Yeah.
I don't know much about it beyond what we learned in class last spring, so it would be interesting.
Yeah,
to know.
absolutely. Right. So I mean, if I think about kind of a traditional brown dye simulation, right? We just start with you have a B sphere and then Q sphere, I think. I'm trying to remember the names, but basically you'll start with your two molecules and they're just kind of randomly placed with one at the center and one on like the edge of some sphere that's been calculated based off of kind of where interactions diminish. And ultimately they can just float freely around based just on this kind of Brownian Gaussian force, as well as the electrostatic forces, right? So the molecules are just, two free molecules interacting wherever they want to go, and eventually either react or go too far away where they're not going to react. And so the goal is to create something that's a little bit more reflective of the sarcomere, right? Where these myosin heads are attached to this big, thick filament, and they're not going to go very far away from the actin ultimately. And so what we can do, or what we've been working on doing, is incorporating springs that just cause a constant force based off the position of the myosin to kind of pull it back if it goes too far away, and hopefully reflect this myosin neck that you see in the thick filament, so that the motion is a little bit more restricted. And so if we go to this spring slide, the idea is to have a series of springs, so kind of a couple of torsional springs, some linear springs, but you can imagine this like just black spring, right, is kind of going to allow the myosin to move kind of linearly along it. But then if you have additional springs, it gives it additional degrees of freedom. And ultimately the goal is to mimic some of these spatially explicit models that have been published previously to create a more realistic environment for Brownian dynamics to occur.
So is the thought to have the spring kind of tether it to one of the spheres so that it can move?
Yeah, exactly.
Okay.
Exactly. So basically there would be kind of an anchor point that'll be relative to actin. If we think about actin as not moving at all, and the myosin head is what's moving, then that is, let's see if I could kind of draw. So we'd have like some fixed, this isn't never going to move. And instead, then we can just have like the myosin head kind of bounce around as if it's tethered or tied to this anchor point.
Cool. Okay. Yeah, super cool. Thanks for explaining.
Yeah, of course. Good question.
So the the idea here isn't completely out of the blue, right? In other words, the original Huxley 57 Crossbridge model is formulated in terms of sort of attachment and detachment probabilities based on the idea back before they even knew what about the myosin head in any detail, that it was sort of randomly moving in a Brownian fashion, and then would have a certain attachment probability that was modified by by how far away the head was from the binding site, assuming that that the head was attached to some sort of spring. So the idea goes all the way back to 1957. And then in more recent sort of incarnations, people have made more explicit models like the one you see where it says Williams 2010, where they have actually, modeled particular spring constants based on particular measurements. So the nice thing here is that even though from the Brownian dynamics point of view, this is just like an ad hoc empirical constraint, it's not an empirical constraint that we have to entirely make up, and there's enough other models that have been developed at that coarser resolution where we can sort of specify what those spring constants are with a certain amount of confidence. Otherwise, it would seem like it is a little bit circular, right? Because you could get whatever you like by just changing the spring constants. But by assuming they don't change with deoxy ATP and using some values that are consistent with previous models, then I think this is kind of a useful way to constrain the Brownian dynamics. Any other questions, Marcus? Okay, so before we go, I noticed that Stephanie is here, and she has kindly volunteered to help organize the we're going to combine our symposium and retreat. I guess at the time we were planning this, we were anticipating it being in person, and now with Omicron surge, it's a little less clear. So I think we should go ahead and plan that it could be in person, but just have some contingencies for doing it remotely if we have to. Hopefully by the time we have the meeting, the situation will have stabilized and we'll have a better way of judging risks. But after all, campus plans to come back in person in a couple of weeks, if nothing major changes. So I think we can still go ahead and plan for an in-person retreat and symposium, but just with some contingency in case we can. I think, we've skipped some meetings because of the pandemic, and I think we've reached the point where we can't keep doing that. So we need to have these, if we have to do them remotely, we have to do them remotely. But I think everyone would agree if we can do them in person safely, then that would be a more enjoyable and valuable experience. But Stephanie, did you want to say anything, or Kelly, or anyone have any questions or want to volunteer to help Stephanie?
Yeah, I don't know, what was the scheduled date again?
I don't actually remember. Kelly, do you remember what date we had scheduled?
For the symposium, it's February 16th. It's a Wednesday.
Wednesday. Okay. Yeah, I mean, I'm very flexible this quarter and I would like to be involved.
Thank you, thank you, thank you. That would be great.
So, another thing we can do is maybe set a time, perhaps January 16th or something, to finalize the date. And if there's the possibility that we could do it in person at a later date, but that it's not going to be possible on February 16th, we might want to postpone it a little bit. I don't think we should postpone it a lot. And, you know, postponing isn't going to solve problems that are beyond our control. But we may want to sort of set a date when we will confirm things so that we give ourselves a little bit of flexibility in case, say, the campus announces that they're going to resume in-person activities on February 20th, then, you know, we might want to, you know, do it after that, for example. But of course, we don't know. So if we could just think about maybe setting a time to finalize the date.
Okay. I do have Joseph and Nate scheduled to do a research talk like this on January 10th, and then Sean and Emma to do it on January 14th. So you can contact me individually if that is not, if those are not good dates for you, and we'll try to work around it. But I'd like to try to do these weekly, and we do have some flexibility if Mondays aren't good for you and stuff like that. But I'm hoping we can stay on track with these if that's okay with Dr. McCullough.
So we used to do them every 2 weeks. And I don't know what people think. Would you, if we do 2 every week, we'll probably cycle through everyone pretty quickly. So, which on the one hand would allow us to catch up, but on the other hand, you know, maybe a little too much as a regular thing. So we could do them weekly for a little while and then, you know, scale back to once every 2 weeks. Is there any kind of preferences on that?
I think that sounds good, to catch up a little bit, but then switch to every other week.
Okay, let's plan on that, Kelly. So we'll maybe catch up, and then once we've sort of cycled through everyone, we can scale it back a wee bit. The other thing is it would be nice to do them in person like we used to at lunchtime and have an excuse to give you a free lunch. But again, that's a little bit up in the air right now. Okay, well, I think that's all I had. And thanks again to Kristen and Marcus for great talks, and, you know, really, you know, just encourage everyone, including the newest students, that even if you only have, you know, very preliminary results, it's fine. This is a good sort of safe place for you to present it and to get some feedback and to get a little practice, and also to invite anyone you want. You know, usually I don't invite the faculty, partly because if I do, they don't come anyway. But I always tell the students, feel free to invite any of the faculty that you, or members of your lab, or anyone else who would like to come. So the meeting isn't really closed. So if you are speaking and you'd like your advisor to listen, or your advisor would like to listen, please do invite them. But we just kind of keep it small and you know, kind of in the family by default. Okay, well, thank you, Kelly. Thanks, Mariella, and we'll see everyone next week, I guess.
Thanks.
Bye. Thanks.
Take care.
Well.